Welcome to Scigest, your fortnightly serving of digestible science from plant and food research.
Kia ora, ko Nicola Sullivan tēnei. Nau mai, haere mai, and welcome to another Scigest episode. Scientists at Plant and Food Research, with support from Lincoln University, are undertaking an Endeavour Fund project to explore whether some grapevines are better at naturally defending themselves from the disease-spreading pest, the mealybug. This week, we are joined by PhD candidate Michelle Thompson, whose PhD is focused on finding out if some grapevines naturally produce higher levels of defence compounds and whether that impacts the mealybugs. Ngā mihi, Michelle. Tuatahi, can you tell us a bit about your background and your career journey?
I actually grew up around grapes. My parents had a small vineyard and made wine as well. So from a young age, I was actually in my school holidays out in the vineyard, helping out where I could. And after high school, I then went on to study at Canterbury Uni, where I completed my master's degree, and I majored in biochemistry. So not long after graduating, I was really excited to land a role here at Plant and Food Research in Lincoln, and I've now been here for over 14 years. Most of that time I've been in the grapes team, which is led by Philippa Barrell. And last year I started my PhD studies, and I'm doing that through Lincoln University with some amazing supervisors there. So even though I've worked with grapes for a wee while in my career, the PhD has introduced something really new for me, and that's working with insects, which I've never done until last year. And it's been a really fascinating area to dive into.
Awesome. Yeah, and those insects, like we mentioned, are the mealybugs, right?
Yes.
So what are these creatures, and what challenges do mealybugs present for the wine industry?
Yes, so as we've said, mealybugs are an insect. They're a sap feeder, and they are a major challenge for the New Zealand wine industry. The biggest issue for the wine industry is that they transmit a virus called grapevine leafroll-associated virus 3, and the virus is known to cause grapevine leaf roll disease. And grapevine leaf roll disease is estimated to cost the industry millions of dollars in lost productivity, and it's actually the most economically significant and widespread disease in New Zealand vineyards. So this is a big deal for grape growers in New Zealand. The impact is really most severe in red berry varieties. So think of things like the Merlot that you're buying from the supermarket. And the disease is known to affect the growth of the vines. It reduces yields, and it also compromises the grape quality, which then can affect the wine. And interestingly, white berried varieties like Sauvignon blanc, you don't see obvious visual symptoms. So in a red berried variety, you get reddening of the leaves, for example, between the veins. And you don't see that in a white berried variety, but the mealybugs certainly still can cause problems. And another one of the issues that they cause is that when the mealybugs are feeding, they excrete honeydew, which is a sugary substance, and that can lead to the growth of black sooty mould on the honeydew. And then again, the black sooty mould affects the grape quality and affects the wine. So if we circle back around to the virus, which is really the main problem, in New Zealand, 3 species of mealybugs are known to transmit the virus. And mealybugs can feed on various parts of the vine. They can feed on the trunks, the canes, the leaves, and the berries. And where they feed depends on the species and also changes throughout the growing season. And for my PhD project, I'm focusing on just one of those species, and I'll be talking about that today. And that mealybug is called the Scigest mealybug. And we chose that species in particular because it's known to feed on the root systems as well as on the above ground parts of the plant. And that's important because we know that mealybug feeding on the roots is a way that the insects can infect newly planted vines. So it's an important mechanism of virus transfer, feeding on the root systems.
What do the mealybugs look like, and do the different species look different?
So the 2 species that are most common in New Zealand vineyards are the Scigest mealybug and the long-tailed mealybug, and they actually look substantially similar. The long-tailed mealybug, not surprisingly given its name, has sort of long filaments on its tail, but otherwise they look very similar. If you saw mealybugs in your garden, for example, they'll look like a clustered together group of insects. They're sort of white or grey in appearance, and that's due to the wax that they form on the surface of the insect. They all cluster together, and what you see most in a colony of mealybugs is the females. The males look substantially different. They're very tiny and they have wings. And I'm definitely working with the females for my project because they feed. They're the ones that transmit the virus.
So the males don't feed?
The males do not feed.
And the females don't have wings.
And the females don't have wings. Yeah, so they do look really, really different.
What are vineyards doing currently to manage the grapevine leafroll-associated virus 3?
Okay, so the current approach to managing grapevine leaf roll disease involves removing infected vines mainly, and also using insecticides to manage the mealybug populations. But that approach is quite expensive because you're having to rip out your precious vines, it's labour intensive, and it's sometimes not very effective either. And one of the reasons that it's difficult is that when the infected vines are pulled out, some roots can remain in the soil, and mealybugs can continue to live on those remnant roots. And from there, the mealybugs are able to move onto other vines or to newly planted vines. If the mealybugs are carrying the virus, feeding on the roots of a newly planted young vine can result in new infections, and so the cycle continues. And there is an added challenge when we're talking about white-berried varieties like Sauvignon blanc. As I said before, visual symptoms are obvious in red-berried variety like Merlot, but they're not in Sauvignon blanc, and that makes early detection of the virus really difficult. And I think all of that highlights a need for better solutions, ones that reduce costs, but also limit the environmental impacts that are caused by current management practices, for example, the use of insecticides.
Awesome. So how did this project come about?
So we already know from some earlier work at Plant and Food Research that mealybugs prefer feeding on some grapevines over others. And to put it simply, we can think of mealybugs being like human children. If you put a meal in front of a human child, sometimes they'll like it and they'll gobble it all up. Sometimes they'll taste it and then reject the whole meal because they didn't like it. And others they won't even try. And if they don't like what's on offer, then they'll either go hungry or they'll go looking for something else. And in the case of a human child, that's a really frustrating situation where they won't eat the food. But in the case of the mealybugs, that's exactly what we want them to do. We want them to move on. We want them to move away from the valuable grapevines in our vineyards and find something else to eat. So then the question became, what is it about certain grapevines that makes them less appealing to mealybugs? What makes one grapevine taste like chocolate biscuits to them and another taste like Brussels sprouts? So researchers at Plant and Food developed a method to test mealybug feeding preferences across different grapevine varieties. They monitored the survival of the insects and the development through the life cycle of insects on grapevine leaves. This was done both in the glasshouse and in the field. And what they found was promising. Some varieties were much less attractive to the mealybugs, and the mealybugs didn't perform as well on some grapes. So what that means is that if they had a low preference, then when they were given a choice, the insects would avoid feeding on them. So when given a choice of two plants, they'll choose the one that tastes better or is more appealing to them for some reason. And when they're given no choice but to feed on the plant that they don't like, they developed more slowly, so they would progress through their life cycle more slowly. And after that finding, the researchers then dug a bit deeper and they wanted to investigate the underlying chemistry. So what is it about those plants and their chemistry that makes them less appealing to the mealybugs? And they found a class of bioactive compounds in the leaves that correlates with reduced feeding. So plants that have high levels of these compounds in the leaves, the mealybugs also avoided feeding on or performed poorly on when they had to feed on them. The particular compounds that we're interested in are known to play a defensive role in other plants, so they might help to deter mealybug feeding in this system. Ross Bicknell, who's now an honorary fellow at Plant and Food Research, applied for and secured funding to explore this a bit further, and that funding also supports my PhD, which I'm very, very grateful for. So the project is funded by MBIE through the Endeavour Fund, and it's called Smart Adaptive Grapevine Rootstocks for a Changing World.
Okay, and what's the focus of this wider MB Endeavour project?
Yeah, so the program is focusing on bioactive compounds that I've referred to. They are naturally produced in high levels in some grapevines, as we already know, and the goal is to understand how these compounds might deter mealybug feeding and how that might work in grafted plants. And just to talk a little bit about grafted plants, because that's important for this project, in commercial vineyards in New Zealand, grafting is a standard practice, and that's where the tissue of two different plants is brought together. So the above ground parts of the plant is called the scion, and that is your wine grape variety. So that might be your Merlot or your Sauvignon Blanc or your Pinot Noir, for example. Then the below ground parts of the plant is the rootstock, and there are a number of different rootstocks that are used in New Zealand. There are about 6 that are the most commonly used, and there's a number of others that could be used. So really the program focuses on the rootstocks. We know that a number of rootstocks are very high in the levels of the compounds that we're interested in. And if we can identify the specific compounds that make the mealybugs less interested in feeding in the rootstock and confirm whether those compounds can protect the entire grafted plant, then grape breeders could potentially develop rootstocks that are naturally resistant to mealybugs, and by extension, we can reduce virus transmission in the vineyard.
And your PhD is part of this project. What's the specific focus of your PhD?
So probably one of the ways I can explain my PhD is to sort of talk about the 3 main questions that I'm trying to answer. So first is, how do the bioactive compounds of interest move in a grafted grapevine? So to answer that, I'm measuring the levels of these compounds in different parts of the plant, including both above and below ground parts of the plant. So looking at the scion and the rootstock of grafted plants. And I'm also looking at variation in the levels of those compounds in different seasons. Second, I'm looking at what is the likely mode of action. And another way of putting that is to say, how do these compounds actually affect the mealybug? What are the proposed ways in which the chemical properties of these compounds could affect the insects? And to investigate that, I extract the compounds from grape leaves and use chemical tests to measure their activity. And there are some published chemical tests that I can use for that. And third is, how do the insects behave when encountering these compounds? And I think that's a really fascinating area, learning about insect behaviour. And for this, I'm studying mealybug feeding behaviour on grape plants that naturally differ in their levels of these compounds. And that helps us to understand whether higher levels of the compounds really do reduce feeding. And again, it comes back down to, can we reduce virus transmission?
Yeah, that mealybug behaviour would be really fascinating, like you say.
Yeah.
And how are the mealybugs being studied as part of your research?
So to explain how I'm studying, I need to talk about the technique that I'm using. It's a mouthful, but to measure mealybug feeding, I'm using a technique called electropenetrography, or EPG. EPG is currently the only method available to measure the feeding behaviours of piercing-sucking insects. So piercing-sucking insects includes mealybugs, and also a lot of EPG work has been done on aphids. In fact, in recent years, it's also been used to study blood-sucking insects such as mosquitoes. And we use EPG because it's incredibly hard to tell what behaviour a sap-sucking insect is performing just by watching it. So visual observations alone just aren't enough. Visually, you can't see the details of how or where it's feeding in a tissue, and that's where EPG comes in. So the concept of electropenetrography, or EPG, is actually sort of similar to electrocardiography, or ECG, or sometimes called EKG. And the idea behind ECG is that we don't need to look directly at a person's heart to be able to see what their heart is doing, how it's functioning, because we can use ECG to record the heart's electrical activity. So in the same way, electropenetrography measures electrical signals generated as the insect feeds. And we can interpret those signals to determine what kind of feeding behaviour is occurring.
Wow, sounds fascinating. You say the mealybugs are sap feeders. How do they feed on the sap?
Yeah, so it's important to understand a little bit about how mealybugs are feeding and what's already known about mealybug feeding before we can understand how the compounds might affect that behaviour. And it's important to understand that mealybugs don't just start feeding straight away. The process of selecting a feeding site, accepting a plant as a host for feeding, and then starting to feed is actually really complex, and it can take hours, lots of hours. I'll go through the phases a little bit. If you have any questions, please let me know. It starts with a phase called non-probing, and during that stage the insect might just be standing still on the leaf in the case of my study, or it might be walking around on the leaf looking for somewhere to choose to feed. It could be using its antennae or using its mouth parts to touch the leaf, And to do that, it's just selecting a favoured spot, and it's also looking at the surface chemistry and the physical features of the leaf and deciding if it even wants to start feeding there. So once the mealybug selects a suitable feeding site, it'll insert its mouthparts, which are also called a stylet, into the leaf, and that begins a phase called pathway phase. And during pathway phase, the insect is actually manoeuvring its stylet between the cells. So it doesn't just go straight down, barging through the cells of the leaf. It carefully manoeuvres between them. And I think that's pretty amazing. I certainly didn't know that before I started studying them.
Yeah.
What it's doing there is it's trying to make its way towards either the phloem or the xylem, which are the plant's nutrient and water channels, and that's where it wants to feed on the sap. And while it's moving through between the cells during this pathway phase, it regularly punctures individual cells. And it's quite well understood in aphids that's probably a tasting behaviour, and in mealybugs it might perform the same function. So they're making lots of little punctures and eventually getting down to the phloem or xylem. Once they reach the phloem or xylem, they can then sit there and ingest for a period of several hours. And really, phloem feeding is of critical importance to my study, and that's partly because we know that the virus, grapevine leafroll-associated virus 3, is likely to be transmitted only during phloem feeding. So that means if a mealybug inserts its mouthparts into the plant and doesn't ever reach the phloem, that's insufficient to acquire the virus. So it won't pick up the virus unless it actually ingests phloem for a reasonable length of time. And in long-tailed mealybugs, it's been found that they need to feed on the phloem for about an average of 1.8 hours in order to acquire the virus.
Wow.
So understanding the fine details of the behaviours and how long they're performing for is critical to understanding the feeding overall.
Cool. You talked about the EPG that you use to look at the feeding of the mealybugs. How do you use that? How do you measure the feeding of the mealybugs?
Yeah, so first, before I could even use EPG to try to compare any plants, I needed to establish methods for using EPG on something that we know is a plant that the mealybugs like to feed on. And so for that, I'm using Sauvignon blanc, a variety we probably all know quite well. And that meant starting by figuring out the best techniques and comparing my data to previous EPG studies of other mealybugs. Plant and Food purchased an EPG instrument to be based here at Lincoln, and it's been quite cool to be able to be involved in setting that up from scratch and getting this working. There's currently no published data on EPG studies of Citrophilus mealybugs, so I've been relying on other EPG studies to help guide the way. And to explain a little bit about the practical side of the setup, first of all, I need to insert a copper wire into the potting mix of a potted vine, and that copper wire is called the plant electrode. I use that wire to apply a really tiny voltage to the plant, and all of that is set up inside a Faraday cage, and what the Faraday cage does is it protects the system from other sources of electrical noise. So because I'm dealing with really tiny voltages, I need to keep out any other sources of electrical signals. So that could be a heat pump in the room, a light fixture, people moving around, so they can all interrupt a recording. So I have to carefully keep a quiet space. Then the next step is to connect a single mealybug to another electrode. That's called the insect electrode, and then that is connected to an amplifier. The mealybug is placed onto the underside of a leaf, which is one of the preferred feeding locations of these mealybugs. Then, when the mealybug inserts its mouthparts into the plant, it completes an electrical circuit, and that creates signals that can then be recorded in real time and showing exactly what the insect is doing as it feeds. So it's just an electrical system that includes the insect and the plant connected to wires and collected to an instrument.
Wow, and then you look at the electrical signal to see what it's doing.
Yeah, we know from other studies what behaviours the mealybugs are performing when we're looking at the electrical signals.
Yeah, it's some really cool methods.
Yeah, and the tricky part of performing EPG is probably attaching the tiny mealybugs to their insect electrode. The insects that I'm using are an average of about 2 millimetres long, so they're very, very little. And the first step involves gently brushing the waxy coating off the insects. And I need to do that because I'm trying to attach an electrode and we want a really nice, clean electrical signal. And as I said before, the wax kind of makes them look grey or white, but underneath that they're actually kind of shiny and brown. So once they're clean, I can then attach the electrode, and the electrode is a very, very fine gold wire. It's about a third of the thickness of a human hair, this wire, so it's very light and very fine, and that's important so that it's not heavy and doesn't disrupt the insect's behaviour. I make a tiny little loop at the end of the wire, and that just sits flat on the insect's body, and that's attached with silver glue. And again, we have to use metal because we want everything to be conductive. Once the mealybug is placed on the leaf, I do observe them for a short period of time, and that's to check that they're able to move freely. And that's really important because we want the mealybugs to be able to select their preferred feeding site and settle somewhere that they really want to be.
This electrode, does it hurt the mealybug at all?
No, I mean, the insects perform normal behaviours when they have this electrode. As I say, it's very, very fine. I mean, I would say the weight of the glue in the electrode would be not perceivable to them, and they are able to wander around freely with it attached to them.
I guess it's like having those ECG stickers on your chest.
Yeah, that's a really good way of thinking about it. That's exactly what it's like. It's just about being able to measure the electrical signals, and we certainly don't want the insects to be upset or they won't behave normally.
Yeah, that's right. It's a really fascinating method of looking at this.
Yeah.
And have you come across any cool findings so far?
Yeah, I mean, I think I have. It's difficult because I can't show you the waveforms here. But the thing that's exciting for me is that I've been able to identify different feeding behaviours by comparing the electrical signals, which are called waveforms, to what's been observed in other mealybug species. And to do that, I'm looking at features like voltage changes, frequency and amplitude, and I can tell based on that information what the mealybug is doing at any given moment. And for example, the non-probing phase, so this is when the insect might just be standing still on the leaf, shows up as mostly flat. Then when the mealybug inserts its mouthparts or stylet into the leaf, there's usually a sharp jump in the electrical signal. Mealybugs will spend a lot of time, usually hours, performing pathway phase where they're advancing their stylet. And during that stage, you see lots of brief downward spikes, and that's believed to be those intracellular punctures. And they usually last about 30 seconds each. So they perform lots and lots of those, these mealybugs. And then once the mealybug reaches the phloem or xylem, the waveform changes again. Phloem feeding begins with a distinct salivation signal, and that normally lasts for a few minutes. And then it transitions into an ingestion phase. And that's the ingestion phase that can last several hours. And remember, that phase is where we think that virus acquisition is believed to occur. And what's really exciting to me is that early on, when I started this, I wasn't able to really resolve the fine detail. So being able to now tell the difference between salivation inside the phloem and ingestion inside the phloem, it's quite exciting to me to have got to that point. And it's really important to be able to distinguish those differences because we know which behaviours are important for acquisition of the virus. I do typically record each mealybug for 48 hours, and I can monitor 4 insects at a time using the instrument that we have. To evaluate feeding on different plants, I need to have at least 10, but ideally more like 20 recordings from individual insects. And then I can analyse all that data and figure out how the insects performed on a particular plant. I'm still at the stage where I'm performing recordings, collecting and analysing data, but I already have some preliminary results for how the mealybugs behave on Sauvignon blanc. And I can tell you that it takes them an average of about 3.5 hours to insert their mouthparts into the plant the first time. So that's just the time that they're spending walking around deciding if it's something they want to feed on. And then around 70% of the insects that I've recorded will ingest phloem, which is important for our study. And that's during a 48-hour window. And phloem ingestion, it typically takes them an average of 20 hours to reach phloem ingestion. So this is a long process. It takes these insects a long time to decide to feed on a plant. But if the insect didn't like a plant, you might expect it to take even longer to insert its mouthparts into the plant, or even longer to reach phloem ingestion, or even better, it never reaches phloem ingestion. To go back to that analogy of the children, it might just have a look at the plant, kind of maybe taste the plant, decide that it's yuck, and just move on. And that's what we'd really love to happen, that the insects decide this plant is just awful and they don't want to eat it at all. Interestingly, a lot of the insects that I'm studying also ingest xylem. It's well known that mealybugs will feed on the phloem and xylem, but in my recordings so far, around 90% of the insects ingested xylem. And while we know that these insects ingest xylem and phloem, it's sort of interesting because they're probably more thought of as phloem feeders. And we think that that could possibly be due to the preparation of the insect. And that's because we need to remove the wax. So the wax could possibly play a role in maintaining hydration in the insect, and removing the wax might lead them to ingest xylem earlier than they ingest phloem, because they feel like they need the watery xylem more than they need the nice nutrient-rich sap from the phloem.
It's fascinating. I never would have thought that tiny little mealybugs had such complicated and time-consuming feeding.
Yes, I know, and it's amazing to me as well. I think the thing, I was talking to some people the other day and they asked me the question, well, you know, if it takes them so long, surely it's actually quite difficult for them to acquire the virus, to pass the virus on to something else. But we have to think of them in terms of the whole population. So, of course, there are lots and lots of these mealybugs, and if you've got a bad infestation, you could have thousands of them. And so at any one time, you will have some insects that are probing and some that are walking and some that are feeding on xylem. And so you can easily see how a population as a whole, if you ended up with mealybugs and virus-infected plants in your vineyard, that population of mealybugs can really easily spread the virus throughout your vineyard if you're not managing the mealybugs through other means. So, for example, using insecticides.
But you can see that if we reduce the numbers, that could have a big effect, right?
Yeah.
And not necessarily needing to eliminate every single one of them.
Yeah, exactly. And it's just about making the plants less appealing. We just need them to prefer eating something else. And so the mealybug population ideally will move away from the vineyard and feed elsewhere. The plants will be unappealing enough that they don't want to feed on them. And the idea is that then we wouldn't have to apply as many insecticides, and that would be great.
Yeah. So you're still working on this project.
Yep.
What is the team hoping to get out of this research, and what benefits could this research have for future wine grape breeding?
Yeah, well, I guess we've talked about it a little bit throughout our chat today, but the idea is that the work that I've done so far, making it possible to measure mealybug feeding on Sauvignon Blanc plants, now gives me the methods that I need to compare feeding behaviours of these mealybugs on grape plants that differ in their concentrations of the bioactive compounds that we're interested in. So that gives me a tool to measure the impacts of those compounds on the feeding, which is great. As I said earlier, I'm also looking at the levels of the compounds in different plants and in different tissues of grafted plants. And hopefully in the future, all that information comes together and will support breeding programs aimed at maximising mealybug resistance in grapevines. And that could potentially reduce the spread of grapevine leafroll-associated virus 3 and improve vineyard health and productivity. And it would be great if we had a future scenario where insecticides weren't needing to be used as much to manage mealybugs in New Zealand vineyards.
Awesome. Thanks for talking with us today, Michelle. It's been really interesting to hear about these little mealybug creatures and their feeding habits and the threat they pose to our bottle of Merlot. So let's hope the research goes well. And to our listeners, thanks for listening to another Scigest podcast. If you haven't done so already, please subscribe and rate our podcast to let us know how we're doing. You can also email any questions or suggestions to scigest@plantandfood.co.nz. Until next time, thanks for listening. Mā te wā.